Okay, so I'm going to get started. Welcome to the UDNF Peers Tell Me More lecture entitled Why is Obtaining a Diagnosis Challenging? Navigating the Uncharted Waters of an Ultra Rare Disease. My name is Christine McGarvey. I'm the director of the Tell Me More lecture series. I am also a UDNF peer member, a UDN participant, and a mother of a UDN participant. Today, we will be discussing the various types of testing used to obtain a diagnosis and their limitations. We will also be exploring the complexity of attaining a diagnosis, and tips to shorten the diagnostic odyssey will be presented. Lastly, strategies to remain hopeful while living undiagnosed will be shared. Just a little bit of housekeeping. If you have any questions, please feel free to put them in the Q&A section, and time allowing, at the end of the lecture, we will answer the questions. So it's with great pleasure to introduce our first speaker, who is also a UDNF peer member, and she will be sharing Felicity, her daughter's story, and how she's remained hopeful. So Nikki Patrick, she lives in the suburbs of Chicago with her husband Josh and her three children, Grace, Brendan, and Meredith. Nikki became involved in the world of rare and undiagnosed diseases after the birth of her fourth child, Felicity. Felicity battled a relentless disease during her short 21 months before she passed away in December of 2020. Nikki is passionate about coming alongside to support families and getting their needs met to better serve their children. And Nikki continues to seek a diagnosis for Felicity while also coming alongside families like hers to help them navigate the process. She has successfully seen two children to an ultra rare diagnosis and has helped many other families understand the process better. Nikki holds onto the hope that one day she will find an answer for Felicity for both her own peace and for future Felicities to come. It is her hope that no parent or family feel alone as she did during Felicity's life. Thank you so much, Nikki, for being here and sharing Felicity's story.
Thank you for having me. I am really excited to do this and to talk more about Felicity and some of the things that we experienced. Is that working? Did I do that right? Okay. So Felicity was born in February of 2019. I had a typical healthy pregnancy. She was my fourth child. Her birth was fairly uneventful. The only kind of interesting things about her were that she was measuring much smaller than my other children had. My other children were all born at 9 and 10 pounds, and Felicity was born around 6. And also she had a lot of just very decreased movement. So when she was born, immediately, we knew something was off. Her breathing was very labored, and there were a lot of just things that we didn't experience with our other children. She spent a little bit of time in the NICU, just getting some breathing help. They thought initially that she had come out very, very fast, and so her lungs were full of fluid, and they thought initially that that was the issue. We were able to take her home after just a few days and thought that, things were going to be normal and okay. And pretty quickly we figured out that that was not the case. She experienced pretty severe weight loss, growth restriction, and I kept taking her into our pediatrician saying, like, it's my fourth child. I know something is not right. Like, what's going on? And they kept telling me, first it was that she had a cold, and then it was that she had very severe reflux, and there were just always kind of like Band-Aid answers. that I just sort of accepted as the case. When Felicity was 6 weeks old, I said, no, something is very wrong. My child is turning blue multiple times a day. She was still under her birth weight, um, despite aggressive, you know, feeding. And it seemed that she was taking in plenty, so it didn't really make a lot of sense to me. And so I took her to the emergency room, and she was hospitalized that time with aspiration pneumonia. And during that time, genetics was flagged. Um, I'm so sorry. I'm also in my classroom. My students are working on a project, but go ahead, Jacob. So anyway, so she was hospitalized with aspiration pneumonia, and genetics was flagged because of some skin abnormalities that were noticed, and also very low tone, the growth failure, and a couple other things. So genetics came in, they discussed things with me, and they agreed that she had some very interesting findings. She was later diagnosed with something called cutis laxa, which is essentially like excess, very loose skin. Um, Her tone was found to be very low. She had a few abnormalities on her MRI that was eventually done. She ended up having just this whole workup of tests. What originally seemed like kind of an innocent pneumonia turned into this whole spiral of thing. We ended up leaving the hospital with an NG tube, and was never able to nurse her again. And we left, and we thought there would be an answer, right? Like, we thought they had run very basic genetic testing at that point, and they told us, go home, feed her with this NG tube, and we'll call you when we have results. Cool. So a couple weeks later, they called, they said, we didn't find anything on this testing, how is she doing? And at that point, she still hadn't gained any weight. So we were readmitted. She was given an NJ tube, thinking that maybe she had some kind of digestive issue, and then genetics came back, reconsulted, and decided to run a whole exome panel. It was at that point that I think things got a little more dire. They realized that she truly was not gaining weight. We spent 6 days in the hospital at that point, where I wasn't allowed to provide any care to her. The nurses were providing all care, and she lost an entire pound under their care. So they thought, something serious is really going on here. They did heart workup, they did various tests, and things kept coming back abnormal, but nothing that unified a diagnosis. So we ended up getting her whole exome done through like a rapid sequencing. And it actually came back with the genetic diagnosis of something called Nemaline myopathy type 8, which is a neuromuscular disease that causes failure to thrive, respiratory issues, a couple other things. And it sort of fit, but typically kids with Nemaline myopathy, especially type 8, present a little bit differently, much more severely. And also have things like club feet and a couple other markers that Felicity didn't have. And the other odd thing was that one of her like mutations, she had 2 from me and 1 from my husband. This has also only been seen in Chinese couples, and neither of us are Chinese. So anyway, so genetics was really puzzled, very confused, and weren't really sold on that being her diagnosis. And actually through the UDN, we found that they're also not sold on that being her diagnosis, and we never really were either. So we, at that point, I said something is obviously very wrong with my child. She still wasn't growing. She wasn't meeting any milestones. And we kind of just went on this odyssey that took us to different parts of the country, took us obviously to the UDN. Unfortunately, our UDN experience was very much impacted by COVID. Felicity was accepted in May 2020 when the UDN wasn't really functioning. We ended up scheduling her finally for early November of 2020. Had a lot of testing done remotely before that appointment in anticipation of that appointment. And then made the decision in late September of 2020 to put her on hospice care. So we did not end up attending an on-site visit at the UDN, but we were able to submit all of her blood work, tissue samples, and all the things they asked for to hopefully find a diagnosis. Unfortunately, December 6th of 2020, she passed away in my arms in our home. And that kind of has become the second part of my journey, which is now trying to not only find an answer for her, but find an answer for the next Felicity. And I think that my most important message today is that there is always hope. We don't know today, but science could catch up tomorrow. And also that having a diagnosis or not having a diagnosis, which for me, I have come to peace with the fact that undiagnosed is a diagnosis. It doesn't mean that nothing's wrong. It doesn't mean that there isn't a problem. It doesn't mean that there isn't a genetic cause for your illness. It just means they don't know today. And that's been really helpful for me as a mother and as an advocate to come to terms with our journey and with, how things have played out, and also holding on to that hope that there will someday be an answer, and there will someday be, because with an answer, sometimes, not always, and I don't even think often, even at this point, can come treatment options or, different roads to go down, because we kind of went, I always say that there was no guidebook. There was no, path. There was no journey. There was just making the best decision with the information that we had at the time. And that would be my second, biggest takeaway from this that I hope that people would come away with, was that you have all this information, and if you don't have a diagnosis or you have an ultra rare diagnosis, you don't always know what to do with the information. And one of the things that has become really, really important for me and a big part of my healing journey in losing Felicity is accepting that I made the best decisions I could with the information that I had at that time. I now do have some different information, but I didn't have it then. And so that's been really helpful for me also. So yeah, if I could just leave you guys with those two things, it's that there's always hope. There may not be an answer today. There might be an answer tomorrow. Undiagnosed is a diagnosis, and you do the best you can with what you have in the moment.
Thank you so much, Nikki, for sharing Felicity's story. And I love that. I'm going to keep that in my heart, that being undiagnosed is a diagnosis. So I think that's a very positive message to share, and healing. So next up, we have Dr. Devin Oglesby, and he's going to be sharing the different types of testing that the UDN offers in trying to obtain a diagnosis. Dr. Oglesby is an associate professor in the Division of Laboratory Genetics and Genomics, Department of Laboratory Medicine and Pathology, and in the Department of Medical Genetics at the Mayo Clinic, USA. He supported the Undiagnosed Disease Network by co-directing the Phase 2 Metabolomics Core with his colleague, Dr. Ian Lanza. Dr. Oglesby's research is centered on translational applications for the laboratory diagnosis of inborn errors of metabolism and rare genetic disorders. His particular areas of interest include developing novel clinical assays and characterizing new biomarkers, and developing methodologies for improving and expanding newborn screening. Thank you so much, Devin, for being here today.
Christine, thank you for the invitation. And also want to thank Nikki for sharing your story and journey with Felicity. My job today from Rochester, Minnesota, where I'm sitting right now, is essentially just sort of to describe, I think, some of the philosophy behind the Undiagnosed Diseases Network laboratory testing approach, and then to give some perspective about why there might still be gaps and individuals like Felicity may fall through those gaps about the approach that's been devised by the UDN. So I'm currently in Mayo Clinic, and I'm going to give a scientific talk. And I apologize in advance if this is pretty technical, but I hope that you can take away from this that there's been a lot of change occurring within laboratory methods and strategies with the UDN, even in the short period of time that the UDN has been in existence as a network, and that has been about 10 years. So the UDN originated as a program within the NIH in Bethesda, but in 2014 commenced as a National Institute of Health Common Fund funded study. And it had three objectives, and it's still continuing that work today, trying to improve the level of diagnosis and care for patients with undiagnosed diseases, increasing and facilitating research into the etiology and linking the clinical and research experts together, and then trying to create this community and then helping to identify and improve options for patients with undiagnosed diseases or diseases that have just recently been diagnosed. In those 10 years and even to today, we see a lot of changes within the UDN, one of which was an expansion of that research network to include quite a few other areas within the US that were not necessarily housed in Bethesda. And this was a map of the UDN sites as they existed until 2024. Today, there are quite a few other sites that have been added to this through a participant mechanism called the NEXO1 mechanism within the NIH that has included, I think, some additional clinical sites. In addition, I think the funding mechanism behind the UDN has changed recently as well. But I just wanted to acknowledge that there were 12 clinical sites and there are more. There's a sequencing core. I helped with my colleague Ian Lanza to direct a metabolomics core that was supporting the network for about 5 years. And then there was model organisms screening centers located in a couple of different parts of the United States. But the structure you could essentially appreciate is something similar to this. Individuals of which a physician or some potential nomination process and committee submitting a potential case for the coordinating center to then help localize those patients in different areas so the travel would be less restrictive. And then clinical care and clinical review, as we heard Nikki described, would be at a particular area where there could be in-depth assessment and then samples obtained for assessing whether there's a sequencing need for identifying whether there might be a DNA variant that essentially could help diagnose that case, or samples sent to the metabolomics core to look at small molecules. And if those two studies, and there are quite a few other studies, would suggest the potential mechanism or model, there is also an opportunity to investigate that further with the Model Organism Center or the MOSC Center. Oops, sorry about that. So how does this differ from a clinical report? And I'll get into this in a little bit more detail, but in clinical genetics today, essentially a patient might be seen by a clinical geneticist and then essentially know what test they want to order and assess whether or not that is the reason behind that genetic finding that they see from the clinical manifestation in that patient. And it's essentially a turnaround that permits there to be an interpretational clinical report submitted to an individual. The undiagnosed diseases space essentially is very unique, right? Because at this point, we're trying to identify conditions which haven't been described in the past. And that requires quite a few additional studies. And this is the laboratory space that I'll be describing in this talk today. These studies are required and also fall into more of a functional study definition and are needed because essentially with genetic testing, there's always this attempt at identifying what might be the cause for the particular manifestation in a particular patient. And this is often done through association. It's very difficult to identify an unknown disease without having an association. In addition, there might be some requirements for a time order to identify whether or not the variation is independent or chronological after another event, and also to demonstrate that it's not spurious, to essentially exclude other variables. And then also, I think most importantly, is to identify a mechanism, a biological mechanism, and that mechanism may be also really requiring some additional studies like a model organism, for instance, to demonstrate that this particular phenomenon that's occurring in a patient really does lead to the pathology that's occurring, and maybe the model organism may help identify that pathology or similar pathologies. But all that requires a lot of testing. Even within the 10 years of the network within the UDN, there have been really quickly changes occurring with the potential to diagnose diseases through a genetic hypothesis, to sequence the inherited DNA from an individual from a tissue sample and ask, is there a variation in that DNA that leads to a pathogenesis and then a pathology in the patient? Originally, and you'll probably see this in your own experience in this space, that there might be a targeted test ordered by a clinician using a certain type of DNA sequencing that is very unique or very specific, and then also asks for additional samples to run another test that may essentially be more general. We heard this earlier about Felicity, where Felicity had a couple of targeted gene testing performed and then came back and asked if they could do a whole exome, and if it wasn't a whole exome, a rapid genome. In each one of these steps, there is a different laboratory phenomenon, laboratory methodology, technology that's being used. And in this example here, I've just sort of highlighted some of the differences that we see so we can appreciate the fact that DNA sequencing has changed really rapidly within the last 10 years. And even before that time, where we were able to query each individual piece of the DNA. And if you can assume that our genomes are essentially like a very large puzzle, and each one of the sequencing methodologies essentially is just looking at a small puzzle piece. By piece and base by base, as they're nucleic acid bases. More recently, there have been sort of massively parallel DNA sequencing methodologies that can look at large sections, about 125 to 250 bases. And more recently, within the last 7 to 8 years, we've seen the capacity to look at even millions of bases simultaneously using what's called long read sequencing methodologies. And this is just describing one of those technologies that I'll go into in a little bit later. Why might that be advantageous? One of the phenomenon is that if you're looking at each small puzzle piece, it's really hard to discern the entire picture that could be on that puzzle if you're just looking at a very minute area, and getting larger puzzle pieces makes it a lot easier to solve. And we can see that in this table right here, where I just sort of described what might be the different genetic testing assays and how they're capable of picking up different types of variants. And some of these don't have the large puzzle piece big enough to see them in particular, but they also have a very inexpensive cost relative to some of the more recent technologies that may be a little bit more expensive. And so, there obviously are potentials to miss certain types of variants depending on what technology is being adopted. And as things get improved over time, those technologies get updated. And one example of this, I think, is actually pretty noteworthy. There was a nice paper that was put out in 2022 where there was a group of individuals looking at a collection of patients that were originally undiagnosed, and they had access to whole exome sequencing, and about 41% of those patients were diagnosed after the whole exome sequencing test, but a good 60 or 59% of them were undiagnosed, and they essentially reflexed those individuals to a whole genome and were capable of getting an additional percentage of patients. But still, the majority of those individuals didn't necessarily have a diagnosis, even after using those two different methodologies. This study also looked to see, after time passed, they went back to the original analysis and just used a different filter or a different approach, used the current literature out in specific scientific press and ask, would a reanalysis provide them with additional information? And they were able to pick up additional patients using a reanalysis approach. And then in addition, looking at new sequencing methodologies, they were capable of actually sequencing those patients again and identifying an additional patient. So even, the methods that we use in the laboratory today are not the same ones that are in existence 10 years ago. And even using the sequencing technologies that we have today, we actually have pipelines that change over time because the literature is updated and our understanding of undiagnosed diseases become a little bit more in depth. And we have additional diagnoses that are obtainable. What were the types of things that were overcome to help identify patients that originally were negative or maybe didn't have a specific diagnosis in those early whole exome sequencings and whole genomes? And those are essentially identified here. Many of the genes that currently were capable later of obtaining a diagnosis were unknown originally. Sequencing technologies became a little bit better. They were able to sequence a little bit more of the entire genome and had better coverage. They knew what they were looking for based on updating the interpretation and filtering process, and also were capable of picking up additional variants that they didn't really have the capacity to do originally in the structural variant process. And I'll give two specific examples about that here. These are two examples of structural variants that were originally missed in old sequencing pipelines and methodologies and then identified after a reanalysis. An example of that is shown on the left-hand side, and you can appreciate something called coverage. If you were to take the entire genome of an individual, fragment that into small pieces, and then sequence in 150 base pairs or 250 base pairs, and then just align those reads above the entire map over the entire puzzle again, you would notice here that there's a space missing in this particular individual, and that was recognized after reanalysis as being a signal of a deletion. And we have areas of the genome that just don't like to sequence well at all. Maybe we haven't defined every individual genome, so we have gaps in these spaces. So it's not uncommon to find gaps in reads across the entire genome, even today, as we have improved the processes, but we still don't have the full definition. And that's one reason why there are current research studies looking at a pan-genome. We know that ancestry and people's genetic origins essentially really influence what that reference might be, what a normal reference might be for particular populations, and identifying as many different sequencing variation that we can in the population will help us build a genome that may be a little bit more reflective of each geographic region or each population or each ancestry. And in addition, in this particular case, what was recognized was that they had a very small amount of signal. They actually had reads spanning that deletion that were called discordant pairs. So they had half of that read was mapped to one part of the genome and half the read was mapped to the other part of the genome. And the signal was very small. It was about 4 reads, out of somewhere where you may have 100 reads. And they recognized that after reanalysis, this is being a signature of a deletion that existed in the population. And we're able to identify that patient after the deletion was found through this methodology. And on the opposite side, you could appreciate the fact that even duplications in the genome, where there are repetitive elements in the genome that may be duplicated through inheritance, that actually can also be just as problematic, where those coverage here that I'm pointing to with my pointer can be quite small. And sequencing that also requires then to understand that deletion signal really does mean that this has got some biological phenomenon. And it's not just that the sequencing instrument is a little bit better at sequencing those reads at that particular area. But there are other reasons why things can go awry when you're looking for a genetic cause. You may be able to sequence things beautifully, and this is sort of an example of all the different variations that can occur that we just certainly don't have a good handle on today and would necessarily not give us a specific diagnosis if we genome sequence someone today. So we can have beautiful alignment. Your puzzle piece can be well defined, and you might think that it fits in its particular location, but actually it could be in the inverse. Because there's some symmetry in that puzzle piece that can go into either way. It may actually be localized in a different part of the puzzle picture, and you could appreciate that if you're looking at blue sky in a puzzle piece, and you don't know where that shade of blue is, and the particular puzzle piece may be fitting all over the place. And this is sort of an example of that, where it's maybe misplaced in the reference, and we just don't know its exact location. But it does a great job. It's perfectly sequenceable. You find it, its coverage is great, everything looks wonderful. Other examples are there might be weak alignment. Maybe there is some poorly sequenceable region that just doesn't look great, or that there are regions that are essentially very repetitive, and you can't tell how many copies there are of that, because there's so many. In this circumstance, the base is A's. There's so many A's there that it just essentially looks like their A's can be mapped all over the place, and you just don't know what to do about it. Other circumstances are there's just too few reads, and I described a little bit how deletion can lead to the too few read phenomenon, but there might just be ancestral occurrences where there's too few reads. And there are some reasons for that. There might be some dropout in particular for the particular sequence. And the opposite can occur. Sometimes you can have too many reads and you're just not finding the variant it's specific itself because it's sort of swamped by normal reads in that area. And that can be due to a variety of different phenomenon. Because there might be duplications in the genome that occur, and that those duplications themselves may be unique or even disease causing. And so there's some solutions for that, and one of which has been trying to get those puzzle pieces as large as you can, and that was where the long read sequencing methodologies that are rather novel and new for the UDN, and I think there's a proof of principle projects that have been ongoing to sort of demonstrate the utility of using longer pieces of DNA to map that area and then try to find where they might be associated and try to get into those areas within the genome. And I mentioned two technologies. The earlier one was using a nanopore, which is a biological molecule that's capable of sequencing using motor proteins, very long fragments of DNA. And it can also do some additional amazing mechanistic descriptions of those molecules. It can sequence the native molecule. It can ask if that molecule is modified through any chemical modifications and keep track of that and identify if that might be different in one patient versus another. And there's a different methodology that's used that essentially takes advantage of the fact that you can make some very large circular consensus molecules that you can read and read them over and over and over again to get a lot of accuracy out of that. And both of these are very valid methodologies that are used for trying to make those puzzle pieces as big as they can. And what does that gain? This is just sort of an example down below of just sort of the additional length that's out there. And we can see that they're not necessarily giving you the same lengths. The circular reads, which are called hi-fi reads, essentially give you, lengths up to 13 kb on average. And then if you use the nanopore methodologies, you can get, 25, 50,000 read lengths. And so the puzzle pieces are a little bit longer, but that gives you access to a greater number of genes that originally were not available when we were first doing whole exome sequencing or whole genome sequencing. They can identify some circumstances where there might be some instability in the genome, and there might be some nucleotide repeats that can expand and contract in different individuals and allow us to count those repeat sizes. And then in addition, there might be some other areas of the genome that are so highly repetitive that you need very long reads in order to measure them and give them the specificity to know where they might be localized in the genome. And those are some of the areas that have been used and are being used right now in the research protocols within the UDN. So I just wanted to sort of put things into perspective here, borrowing a nice figure that was from Johannesson's paper here from 2023, but to just sort of, identify the fact that in the UDN, there's the recruitment, the collection, the medical records, the interpretation of variants based on clinical features, whole exome, whole genome sequencing, the reanalysis process, and then the phenotyping that may occur while you're in a UDN visit. But then in addition to that, there might be some additional methodologies that are being used that are not necessarily available in every clinical office, but maybe only available at the UDN. And one of them was metabolomics, and that's what I'll describe here. And essentially what that is a way to sort of look at the entire spectrum of molecules that may be present in a certain type of sample, and that can be urine, plasma, or CSF. And that uses a mass spectrometry that essentially measures the relative intensities of all the metabolites and lipids that can be identified in a particular method, and then does a comparison of that metabolite against either first-degree relatives or can be also a reference population. This is essentially a way to just sort of get a snapshot. And it's used in two different forms within UDN, and it's often used in two different ways within the clinical space. Clearly, there are so many metabolites in plasma, urine, and CSF that, the 5,000 essentially features that you can identify account for just most of the metabolic processes that change quite rapidly in our bodies, whether we're fasted or fed, or awake or sleeping. And even during the circadian rhythm during the daytime, we see huge numbers of fluctuation of changes of small molecules everywhere. In the UDN, we've really focused on trying to quantitate and measure particular analytes, and then also use an targeted approach to get a better understanding of the hypothesis generation that might be available if we just take a snapshot of every possible molecule signature that we can pick up in a particular sample. These two ways are, I think, quite beneficial. One of which, in the targeted methodologies, we were able to pinpoint certain areas of biology. to sort of ask very specific questions and try to answer very specific hypotheses. And sometimes those hypotheses that we're actually trying to answer is to negate that option, meaning this patient clinically looks like they have a disorder that may be related to how their body is capable of decorating proteins with sugar molecules or glycosylation. And the question would be, do they have a glycosylation defect? It's very difficult to do an untargeted approach with that question in mind when you just need a very specific answer, yes, no, versus the untargeted methodology, which we often have used for hypothesis generation. I'm not certain what this patient's metabolic profile is. Can the untargeted methodology provide us with something like a heat map that allows to assume that there might be some pathways that are impacted in that patient that we can then dig further genetically to identify whether or not there might be something supportive. In the glycosylation area, we have an area of our laboratory that's very uniquely situated to do the type of analysis, and had two different methodologies that we're looking to see what are all glycans impacted on every single protein, or are there specific glycans impacted on specific proteins. And there are certain types of disorders that can help identify whether or not we might be able to pinpoint what genetic gene or what gene to look at in further detail, depending on the glycosylation profiling. In addition to that, though, I wanted to just touch briefly in the next couple of minutes about transcriptomics, because I think this has been an area that you may have seen in the scientific press growing and growing, specifically looking at the messenger molecule that comes off of DNA that is often transcribed into RNA, and measuring those RNA levels to ask, can that help guide us to identify what might be an associated genetic defect? And so transcriptomics is an area I think that is seeing a lot of growth and is also very potentially powerful. And I won't touch a little bit on the methylation signature, and methylation was one way in which the DNA molecules are being modified. And that is an area I think that is also going to be very helpful to solve some additional cases. But in the transcriptomic space, there's a couple of different ways in which the UDN has looked at transcriptomics. One of which is to look at the expression of a particular gene and see if the proband or the affected patient has a different level of expression versus a bunch of controls. Another powerful mechanism the UDN has looked at transcriptomics is to actually try to identify the particular transcript off of a gene that might demonstrate some mechanism for a variant. In particular, there might be some types of DNA variants that lead to skipping of different parts of that gene, and the UDN has worked really hard to identify whether or not they can pick up the aberrant transcript and identify that, yes, there's an exon skipping occurring, and they can find the transcript molecule that shows that. And then in addition, that's different than what you see in control populations. And there are several different mechanisms to do that, but aberrant skipping can be very difficult to perform because you really need to be able to be sensitive to identify that skipping processing molecule. And sometimes those things are so rapidly degraded, or they're very tissue specific, that you can't even use them in a blood sample or in a tissue sample that's accessible at that point in time. So a nice description of the use of the RNA expression differentiation, I think, was published, or actually it's still in preprint, if I'm not mistaken, done by a Stanford group. And in particular, using the long read technology, they were capable of getting into different parts of the genome that they weren't really capable of counting. One of which was tandem repeats, or the copy number of a repeat expansion that was occurring. And they had a group of patients where they identified whether or not there might be a structural variant. So the genome itself was in an orientation that was not necessarily as expected, like as an inversion. And they also looked for duplications and deletions, areas that were so big that the small methodologies of small read sequencing were not capable of picking up in particular areas. And then from that, they then used cultured fibroblasts or whole blood from patients and identified through the RNA sequencing, or looking at the transcript expression, where there might be some expression outliers, and try to tie both of those data points together to pinpoint new genes that were not recognized as being disease causing. And an example of that was shown here, where they had an affected sibling and a proband whose expression outliers were so far out from the median they'd seen in their population that they knew that this was a transcript they needed to dig into further. And in addition, they went back to the genome or long read sequence to the same DNA and found that the repeat copy number in that particular gene was expanded, and demonstrated that that expansion probably changed the expression of that gene. And then in addition, they had another proband where they had found leverage first where the transcript expression was really, really low in fibroblasts. And they thought that there could be a loss of function of a gene. And using the sequencing methodologies, realized that there were two different deletions in areas of the genome that were just outside but adjacent to the gene of interest. So that probably impact the expression off that gene. Again, not knowing that the gene itself was involved, was a disease causing, but at least there is a mechanism for why the expression is low. There's a deletion in the gene. Maybe that gene, essentially through additional studies that would be required, functional studies that would be required to show that that gene is actually leading to disease. So this, as I end right now, and I'll pass this on, I just wanted to just sort of give everybody what is the philosophy, I think, regarding laboratory testing within the UDN and why there might be undiagnosed cases today. We've seen short read sequencing and the increased use of long read sequencing to try to solve some of those very difficult to sequence regions and also to identify variants that are not easy to find. We've heard about RNA sequencing and using expression outliers as trying to identify new regions that may be impacted by a genetic alteration, aberrant splicing, where there might be some skipping of some parts of the gene. There's also allele-specific expression. Sometimes looking at the RNA identifies that the diseased causing allele is the one that's overexpressed in a patient versus what might be seen even if the particular variant is not found at a high level in that patient. I also mentioned the reanalysis study, and I think this is extremely important. I think the computational changes that are occurring right now and the updating of pipelines is extremely important. Case matching, I didn't even describe, but there might be an individual out there who has a hypothesis that this particular variant is disease causing, but they need to go beyond the association and start to look for other cases where that is occurring as well, so they can make sure that this isn't necessarily a spurious finding, and that additional data, like additional probands, would help do that. And case matching, it also has occurred within the last 10 years, has expanded dramatically. Metabolomics, methylation profiling. Then in addition, there might be some other phenomena that necessarily aren't impacted specifically by our genome itself, but immunological changes that may be occurring in a particular patient that leads to a phenotype, and proteomics being the investigation of a lot of different proteins and different samples that may help identify whether that is part of the mechanism of a particular patient. And then likewise, the interaction between functional studies and a diagnosis using either in vitro cell lines or in vivo model organism systems are extremely important in sort of completing that whole picture, especially in the discovery stage. So I just wanted to end right here and say thank you to everybody, and I'll pass this on, Christine. So sorry for going over time.
Thank you so much, Dr. Oglesby. I think that was fascinating, and also filled me with hope for me and my daughter getting the diagnosis one day. I remember when we found out that our whole genome sequencing was negative, it was kind of like, oh, is that it? And I think, you know, we hear the word whole and we think if it's negative, then it's not there, but that was really fascinating. So thank you. Our last speaker is Nicole Wally, and she is actually an ex officio member of UDNF Peer. And before I introduce her, I just want to say that the Q&A function does not seem to be functioning. So if you have any questions, please just put them in the chat section. So Nicole is a clinical research coordinator in the Department of Pediatrics at the Division of Medical Genetics at Duke University. She received her MS in biology at Duke University and is also a trained phlebotomist. She has been a study coordinator at Duke since 2009 and has been the lead study coordinator for the Duke UDN clinical site since 2015. Nicole works with the UDN staff and physicians to coordinate and execute participant evaluations, and she participates in multiple UDN working groups. Outside of work, Nicole enjoys coaching soccer, walking her dogs, and working in her garden. And Nicole will be explaining a little bit about why it's so challenging to obtain a diagnosis and also tips to help shorten the diagnostic odyssey. So thank you so much, Nicole.
All right, thanks everybody. Thank you for having me. And please feel free to put some questions in the chat or stop me if anybody would like to discuss anything more. In my talk, I'm going to try to hurry so we do have some time for some discussion here at the end. But mostly what I think I want to address is in our work with PEER, we've identified some frequently asked questions of things that tend to be challenging, both with UDN participation, with the steps after UDN participation, and challenges during the entire process of the UDN diagnostic odyssey. So often a frequent question we get is sort of, what is the difference between a clinical test and a research test? Very briefly, a clinical test is what you think of when you go to the doctor's office. They order a blood test or something else to be done. These are tests that are done in the hospital. They have standard turnaround times. These turnaround times are published. You can expect them to go by the books. And specifically with regard to the timelines just are different in the research world. So our research tests don't necessarily follow these kinds of clinical procedures. The turnaround times are rarely published. And because they're done in research labs, generally labs are trying to work as efficiently as they can, which sometimes means holding samples until they have enough to fill up an entire sample plate or an entire array to make sure that they're using very respectfully and conservatively the resources that they have available to them. The utility of these tests also is a bit different. So in a clinical test, you will get your results. They will go into your medical record, and these results can be used for making any kind of medical or healthcare decision that you and your doctor see fit. Research tests, again, are a bit different in that they might lend weight to a specific diagnostic theory that we're working on, but in and of themselves, they are not necessarily a piece of medical data that should be used without any additional evidence for making your healthcare decisions. Also, these research labs are not certified by any authority. And so getting these kinds of results entered into the medical record to affect care can sometimes be difficult and can require extra tests to be done so that we have the kind of medical grade data that we need to be able to make healthcare decisions together. And finally, the way that these tests are billed or what they cost can be different. Because clinical tests are done in the hospital will issue a bill. This will go to an insurance company. Patients may be responsible for their copay or any uncovered cost. Research tests are not billed that way. Generally, patients may not get a bill for these things, but remember, that doesn't mean that they're free. So all of these tests do cost something to perform. Now, one thing that can be confusing is do remember that researchers do clinical tests for research studies or for research purposes all of the time. So as part of your UDN participation, you probably got several lab tests during your evaluation that are clinical tests, but we did them as part of your UDN evaluation. So that's a clinical test as part of a research study. The way that these are billed can be highly variable. It'll depend on the study, and it can depend on your clinical site. So these rules are not hard and fast. There's a lot of gray area and a lot of overlap between them. Okay, another question that we have come up against recently is with respect to these various tests, especially the research tests and long-read sequencing and transcriptomics or RNA sequencing that Devin was talking about in his talk. Which is, I know some cases have gotten this test. Is my case going to get this test also? And the truth is that because we do have limited resources in how many of these tests we can do and the amount of dollars we can devote to these tests, we do not do all of these various research tests on every single case. And it is up to the investigators at each site to consider the strengths and the limitations of each technology and the clinical history of each case. And what we do is we try to base our decisions so that we can maximize the likelihood of success of these tests. So we will apply a certain technology where we think it is most likely to be fruitful. One example of that is with the transcriptomics or the RNA sequencing. We've come a long way, but there's still a long way to go in being able to figure out how to do the best analysis of this kind of sequence data to give us or point us towards diagnoses. But one place where it actually can be very useful is if we are looking to determine functional effects of a specific variant that we already know about from exome or genome sequencing, or if we're looking in just a subset of genes that we think might be likely to be associated with a participant's phenotype. And then we can ask a very much more direct question of the data to get more clear answers. And that is one way that we use our resources conservatively to try to maximize the amount of information we're likely to get from this test instead of doing a lot of RNA sequencing or long read sequencing, which generates a lot of data. And we're not really sure in a lot of cases how to make the best use of that data at this time. One other question that we've talked about with Pierre a lot is, exomes and genomes come back with variants of uncertain significance, and this happens quite a bit. And so the question is, well, but what are you really thinking? And the truth of the matter is, we don't know. These are of uncertain significance, and uncertainty can be hard, and it's hard for us too. But we have to be very careful that we're not giving out incorrect diagnoses or incorrect information, because that can be harmful also. But what we can do is we can always talk about sort of the evidence we have both for and against this variant being the cause of participants' phenotype. Your genetic counselor or your UDN study investigators can talk about that with you so that you can better understand why this might be the answer, but also some reasons we're not quite sure that this is the answer. We can also discuss what other next steps might be available to help give us more information for or against this variant being the cause of participants' phenotype. The best questions to ask your clinical site or that your clinical site might discuss with you are things like, is case matching helpful? So if I find another patient somewhere that has a variant in this gene, will that give us more information? Can we do some kind of animal modeling? The UDN does have access to some model organisms. Would those be helpful to sort of determine whether or not this variant or this gene is likely to be disease-causing in my case? Or is this a variant that will lend itself to transcriptomics or RNA sequencing so we can try to figure out what the function is and what other basic lab tests might be able to be done to help us figure these things out. All right, and then a big question is, if your sequencing come back negative, will you ever get a diagnosis? And the answer is we sure hope so. The average time to a diagnosis, don't anybody quote me on this, this is some quick and dirty calculations I did with the data available to me in Gateway. But the average time to diagnosis looks like it's between 1.5 and 2.5 years, but the longest time is over 8 years. And in making this presentation, I could think of at least 2 cases off the top of my head that just took a really long time. In one case, we had a variant that we had been sitting on. We knew about it since 2015, but there was, like I said, there was some evidence in the for basket. This looks good, but there was also some evidence in the against basket. We don't know. It had never been associated with disease, and we've never seen any other patients. Finally, in 2022, so that's about 7 years later, we learned of some additional cases, did some additional research, and we were finally able to make this diagnosis and provide it to the participant. And similarly, we had exome and genomes that were completely negative for another case, not even any single variants. And just with some new phenotypic clues, we were able to perform some very specific and targeted genetic testing. And that led us to identify a copy number variant that just was not identified on genome sequencing because of some technical limitations of the sequencing done at the time that it was performed. So it was really just bad luck that it was missed in 2018, but we were finally able to identify it last fall in 2023. . All right. And if you've had all the tests, but you still don't have a diagnosis, why is that? The sad answer is that most of our participants have not yet gotten a diagnosis. So only about 30% have come through their evaluations and walked away so far with a diagnosis in their back pocket. There are lots of reasons that these cases are so hard, and I'm sure each and every one of you knows that your case is hard, because if it wasn't hard, we'd know the answer already, right? And so there are a lot of limitations with many of the genomic technologies that we are relying on. So, you know, the whole exomes and the whole genomes, as Christine aptly pointed out, the word whole is right there in the test name, but they just don't find everything. So we know that there are limitations to these technologies, and there are genetic variants that these technologies do miss, because we don't know how to construct the analysis in a way to pick out all of the different things. It could be that disorders are caused by 2 or more genes working together. And when these gene variants in isolation don't cause a phenotype, but in combination do, that's really difficult to tease apart, especially when we are counting on more than one person to have the exact same combination of very rare variants. That's going to be very rare. It's going to be very hard to know for certain that something like that is what is occurring. Environmental exposures can be hard. Population-based studies tend to be the best way to try to tease apart different environmental exposures, and those are just not something that this particular research study is set up to do very well. And environment and gene interactions are going to be even harder. Again, they require multiple people to have the same rare genes and the same rare environmental exposures, and for us to know about all of them. And that's going to be very difficult for us to learn about in a short amount of time. Epigenetics is sort of one of these other parts of genomics that we've known about for a long time, but similar to RNA sequencing and long-read sequencing, our ability to do broad analyses of epigenetic data and then to apply those to novel phenotypes is still limited. There's still a lot of work to be done to make the best use of this kind of technology that we can. And then finally, uncommon presentations of other known disorders. These are just things that are hard to determine in isolation. You know, to have the best evidence, we really need to have multiple people with the same rare presentation, and it does happen, and it can happen eventually. It's just hard for it to happen on demand or in a small amount of time. And then just some common questions and challenges that patients have often after their UDN evaluation. One is, how often will your clinical site contact you after the evaluation has ended? The shortest answer is, once you have your results, and we've talked about, what the next steps might be, if we know of the next steps at that time, there is not necessarily a schedule where the UDN clinical site can contact you. We just are not staffed to have that kind of ongoing contact and interaction with every participant. But we are always happy to hear from our participants with updates or questions. And if you ask questions, then you should expect a response in reply. We will, however, in all instances, contact you if suddenly 8 years later, we have a diagnosis. So those are the kinds of things that we will definitely initiate contact for, although we may not initiate contact for regular check-ins or to see if there's anything new that we should know about. But at the same time, if there is something new that you think we should know about, by all means, please contact us. If it's been some time and you're not sure exactly who to contact or how to get in touch with your clinical site, the UDN Help Desk knows how to get in touch with everybody. And so you can always send an email to the Help Desk email or call the Help Desk staff, and somebody will help you figure out who it is that you should contact. And that was all I had for today. So if we want to, if we have time for questions, that would be great.
Thank you so much, Nicole. It is on the hour, but I did have one question. why is it important for the participant to update their clinical site with medical information? Because that could be used for, what could that be used for, I guess?
Yeah, so that's a really good question. So I guess most generally is there are lots of reasons that we have sort of variants of uncertain significance or different things that we might be able to see in the genome and we're not sure what to do with. And one of the things is, oh, this variant looks interesting, but the gene it's in doesn't match the phenotypic presentation of this participant. So as your phenotype maybe progresses or changes, or you have new symptoms, it may let us know that something that we had already talked about or known about that we didn't think was a good match for you because it didn't fit your phenotype. Everything we knew about the gene or the disorder seemed a bit off or not quite right. Maybe now it does. And so maybe we didn't think that shoe fit, but in fact, that shoe fits a little bit better now. And so maybe that's some updated information. And, in the case that I talked about very, very briefly, it was a patient who had multiple MRIs in the past and they were all read out as basically normal, but they got a new one and the radiologist thought he saw something. And what he saw turned out to be iron deposition. And the iron deposition lends us to think of a very specific set of genes. So then what we were able to do was look at those genes, and we found in one of those genes a deletion that was not called in the analysis of the genome data. So it was just a tech limitation that we didn't know about, and because we hadn't previously had any indication that we should be looking at the iron accumulation genes, the brain iron accumulation genes, we didn't know that we should look very closely at those genes. And then when we did, the diagnosis was sitting right in them.
I think that's an important takeaway for all undiagnosed patients, is that I like to think of it as like it's a million-piece puzzle, and each little new symptom or different diagnosis could be another piece of the puzzle that could actually put it together. So it's very important to maintain contact with your clinic site. So I just want to say thank you to all the speakers for volunteering their time, and thank you for all of us who joined us on the call. And we will have another event on August 22nd. It will be a book launch about obtaining resources for disabled children. So I hope you will join us then. Thank you, everyone.